All right, so now we're going to move into more exciting things, in my opinion, in terms of chemistry and physiology, which is the four biomolecules that are essential for life. Does anyone know what those are? Can you list them? What are the four biomolecules of life? Or carbohydrates, lipids, proteins, and nucleic acids. Right? Sometimes we say sugars with carbohydrates, but we'll keep it carbohydrates so we don't get confused. Right? So all four of these things are essential molecules for living organisms. They all have distinct functions and functionalities, hence why we call them biomolecules. So we'll move into them. So carbohydrates, I feel like everyone knows what a carbohydrate is, especially in terms of like if you've played sports. But this is really a lot of where our energy storage comes from, and that we ingest, right? Potatoes, starches, which is, oh look, it's a carbohydrate, a starch, right? Multiple sugar groups together. A lot of our food is based on carbohydrates, right? Because they pack a lot of energy. So in terms of biomolecules, though, we can classify carbohydrates into kind of three different classes. One sugar or monosaccharides, which is just one sugar group, such as glucose. If you have two sugars together, they're disaccharides or sucrose, figure A here. And then if you have multiple sugars together, we call them polysaccharides, and those are like starch and glycogen, right? And it's a little fuzzy here, but you can have multiple branching, multiple glucoses together, right? Lots of energy stored up in there when we digest it and break them down, right, via catabolism, right, to release the energy to make ATP and so forth, right? So this is carbohydrates. It's a big source here. It's also used in a lot of other things in cells outside of energy, right? And so we'll try to highlight those in the cell biology chapter, which is in the next chapter, in chapter 3. Now, lipids is another biomolecule. These are made up of fatty acids, right, chained together, and it can form different types of other molecules: triglycerides, saturated fatty acids, unsaturated fatty acids, steroids, and I always pronounce it weirdly, but eicosanoids, right? These are different molecules made up of lipids. So then you may be asking, like, what is a lipid, right? So an example of a lipid, a triglyceride, right? So you have fatty acids, which is this chain of carbon, hydrogen, and oxygen with a hydroxyl group, the OH group, right? Paired in the backbone here. So these are our fatty acids, long chains of them, right? Paired into a glycerol group. This is a triglycerol molecule, right? So three glycerols, hence triglycerides, paired with their fatty acids, right? So this is mainly this form of lipid, mainly used for insulation and energy source, right? It's energy dense. So there's different ways that we store energy. Triglycerides is one of them, right? So we could use enzymes here to pair the fatty acid to the three glycerols here to make a triglycerol molecule. Phospholipids is a big one in biology and cells because they form the phospholipid bilayer, right, which is the cell membrane, right? So where one fatty acid is replaced with a phosphate group, right, at the head. So you have one end, which is hydrophilic, which means it loves water, and then a nonpolar end, which is hydrophobic, so it's scared of water, right? There's our polar, nonpolar coming in, right? So polar, nonpolar. Water is polar, so like with like, right, to nonpolar, which is the hydrophobic, right? Have a nice little cartoon here, right? Another way of saying that they are both hydrophilic and hydrophobic is they are amphiphilic, so they are both love and hate water, right? Amphi, which means both. In this case, so they have a polar head and non-polar tail. So they love and hate water, right? It's a love-hate relationship. Excuse me. Another form of lipids that we care about is saturated and unsaturated fatty acids. So saturated fatty acids is a long chain of carbon, hydrogens, and the carboxyl group bonded together in a single chain of single covalent bonds, saturated. So basically what this means is all the carbons are saturated with as many hydrogens as they can bind to, right? Because carbons like to make 4 bonds. 1, 2, 3, 4, right? Which means that each carbon can bind to 2 hydrogens most of the time, unless you're at the end. Then it's 3. Then it's another type of molecule, but excuse me, again, not going to worry about that. Unsaturated means there are double covalent bonds mixed in here, right? And if you noticed here, then these 2 carbons in a unsaturated lipid can only bond with 1 hydrogen because the extra bond is with the carbon next to it. This forms kinks in that fatty acid backbone. What this does is it generally makes the fatty acid a liquid, right? Which also means that they are more particleized, easily particleized in water to form droplets. Hence why unsaturated fatty acids are better, because they won't stick to or become solid at room temperature because of these kinks in their chemical backbone. Now, trans fats, we've probably heard of these, are unsaturated fats that are alterated to become saturated. So what they do is they kind of twist the backbone kinks here so it straightens out the molecule, and then they can become solid. So that's why these trans fats are not good, right? And why we suggest more saturated than unsaturated fats. But they're both found in nature here. Other things, so lipids, eicosanoids, these are molecules derived from fatty acids. They're important in like those feedback loop systems, regulatory molecules. They're very kind of minor in terms of what we'll learn here, but they're thromboxanes, leukotrienes, the word's always hard here, prostaglandins. These are just kind of involved in, I think some of these are like digestion. There's a whole broad class of them. These are just examples, but they're more signaling related. So they help more signal than anything. So we're not going to worry about them too much. But one important thing to highlight here is that we need fat in our bodies because there are vitamins that we need, like A and D, that are nonpolar. So they need nonpolar molecules to dissolve them, right? So we need fat. So these are fat-soluble vitamins like A and D versus water-soluble vitamins, which are polar molecules based like B and C. Steroids, probably heard of steroids before. They form ring-like structures, which is pretty interesting. So like cholesterol, we think of that in terms of like blood levels of cholesterol, but cells actually have the basic chemical of that. In the cell membrane, it helps the cell membrane, be soft or stiff. It's a nice way of putting that. Steroids can be produced in salts. Reproductive hormones are steroids, ring structures. Vitamin D is a steroid. So there's a lot of different examples of lipids that are ring-like structures that we don't consider lipids naturally, but they are, just because they're just a different structure here. So they're just carbon-based ring structures. Examples: cholesterol, testosterone, estrogen, vitamin D, cortisone, so forth like that. The third class of biomolecules, proteins, second to most important in my opinion, right? Because proteins are really the workhorse of the cell, the workhorse of our tissues and organs. They are the things doing a lot of the work. And they are made up of amino acids. So these are molecules, different types of molecules based upon what our special groups, R groups are attached to them. So that make them unique and have different functions and different properties. We're not going to go into them because that's organic chemistry, and you guys are not organic chemists or going to medical school or like me and was crazy and had to take organic chemistry. So you just need to know amino acids are the building blocks of proteins. There's 20 of them, and they have different functions, and their order of them when they are put together matters. We often call them polypeptides because they form a special bond called a peptide bond. This is just a covalent bond between amino acids formed by a dehydration reaction. Proteins have structure to them and a sequence to them, right? Because there's 20 different types of amino acids. Their order matters because that determines their structure, which then structure determines functions for proteins, right? And function is really important for proteins. Therefore, sequence determines function, and that's why we care about their sequence. It's just related to our genes. So primary structure of proteins is called the amino acid sequence or polypeptide, right? So many peptide bonds. Now, when you take that primary structure, that chain of amino acids, and start forming a 2D structure, we call that a secondary structure. And what that looks like is two different types of 2D structures, either a pleated sheet structure or an alpha helices structure. So beta sheets, alpha helices. So those are the two secondary structures. When you combine those secondary structures folded together, you make a tertiary structure, which is the next slide. One important thing I want to highlight here is tying back in the pH concept here is if you have a lot of hydrogen ions, so very low pH, that can denature a protein. It breaks the hydrogen bonds and other bonds in the proteins. It breaks down their structure, which affects their function, right? So that's why pH is bad. It breaks down proteins. It denatures them. Heat can also do this. So that's why, like, one of our sterilization techniques is heat, because it will break the heat up, break the proteins up, say in the bacteria. So a tertiary structure is where you start large-scale folding all of the pleated sheets and the helices structures together. And this is all still on one single polypeptide chain, right? So there can be helices and sheets all on one single chain, as the figure shows here. And there's different regions that can fold together and form a quaternary structure or a more 3D structure. But you may be asking, well, how does the protein fold into a 3D structure? Well, the polar regions and non-polar regions of each polypeptide chain determine the structure, the tertiary structure. So when there's a polar region inside the polypeptide chain, it wants to be exposed to the water, so it will stick outside the 3D structure, versus a non-polar region, which hates water, will fold and stay more inside of the polypeptide chain structure, of the tertiary structure. So water, again, is really important here and determines protein structure based upon the amino acid sequence, which then, structure determines function. So we care about where the polar regions of these polypeptide chains are and where the non-polar are. There are certain things called like a domain, which is sections of a protein that perform a very specific function, but we're not going to be worried about that, but just so you're aware of that. Now, quaternary structure is where you just combine multiple tertiary structures, which, remember, tertiary structures are just 3D structures of a primary polypeptide chain, a singular polypeptide chain. So quaternary structure is just basically multiple 3D folded polypeptide chains put together to form a function. So I have a question here. So what is the special class of proteins that we have already talked about? And we talked about this in this chapter and the previous chapter. Anyone have a guess? Yes, enzymes, right? So enzymes are a class of proteins that are vital to proper functioning. And here's how they work, right? So remember I said proteins catalyze or speed up a reaction by lowering the activation energy, right? How it does this is it brings those reactants close together in physical space, so in proximity, right? So it has a three-dimensional shape, which means it has a site where these reactions are occurring, which is called the active site. So where the reaction occurs in the enzyme is called the active site. There's how the reactants identify specific enzymes or vice versa. So enzymes only react with very specific reactants. So how do enzymes and reactants know to match one another? There's two lines of thinking. The science is now really shown that number 2 here is the most accurate, but it's the lock and key. One is the lock and key model, where it's just like there's a key to a very specific lock and only one lock, unless you have the master key. The reaction only occurs to one reactant in an enzyme, right? At the active site, has a very specific shape to the reactant in it. The induced fit model, which is the most accurate, is what we find is that the enzymes will change the shape of the physical shape of their active site to fit the incoming reactant. It's still a very specific reactant, but it can mold and fold its active site to better fit the reactants coming in to catalyze that reaction. So the number 2 is the most accurate from what we found. There are other things that enzymes bind with. One of them is a cofactor. These are molecules that they're not proteins, but they bind to the enzyme to help the enzyme perform a function. So they're kind of required for the functionality. One of these cofactors is like enzymes. They are called coenzymes because they help the enzyme more specifically. They're required for the enzyme to work. So that's an example of proteins, which is our enzymes, right? Enzymes are an example of proteins. The last biomolecule example that we have here, and we'll end this chapter on this, is nucleic acids. So nucleic acids are composed of nucleotides, which are deoxyribonucleic acids, or DNA, and ribonucleic acids, and adenosine triphosphate. So ATP is a nucleic acid. Right. So nucleotides are kind of grouped into 2 different groups here. One is a pyrimidine. One is a purine. All that means is one has a single ring structure in the chemical. One is double ring. So you can kind of see that here, right? Purines have 2 rings. Pyrimidines have 1 ring. One way to memorize the difference between pyrimidines and purines. Pyrimidines are pirates because pirates have a singular hooped earring. Purines are like a married couple. They're pure. They have 2 wedding rings. Wife and husband have both a ring, so there's 2 rings there. They're pure. It's just Dr. Ranahan in the bio department. I was a student of his back in the day. That's how he jokingly said to memorize them. But you memorize it however you want. It's really up to you. But there's 2 purines, guanine, and adenine, and then cytosine, thymine, and uracil are all pyrimidines because they all have singular ring structures. Adenosine, ATP, right? It has an adenine group in it attached to and forming adenosine here, right? Which then is bound to ribose sugar. That's what this is. Which then you add in your phosphate groups, 1, 2, 3. So 3 triphosphates, adenosine triphosphate. So this is what the ATP actually looks like. Now, in terms of our nucleotides, they have like the nitrogenous base here, so you can see the nitrogen, right? And then as well as a phosphate here, which is so 5 carbon-based sugar with a nitrogenous base and a phosphate group is really what we say with nucleotides. These pyrines and pyrimidines here are nitrogenous bases, and then we have other sugars to form the sugar for deoxyribose or ribose. Or we have an image of, I think there's an image of that in the textbook. But so we have DNA, deoxyribose, nucleic acid. Why do we care about DNA? Anyone? It stores our genetic information, right? So what is our genetic information? It produces proteins, which proteins provide the functionality, right? So that's why we care about DNA, our genetic code. Now there's this other molecule called ribose nucleic acid, and why do we care about that? Well, it is kind of an intermediary copy of the DNA, which then is used to transform the DNA code into proteins, right? In chapter 3, we will talk about gene expression towards the end of that chapter, but we care about these two molecules because they are basically how our genes, our DNA, our genetic information is stored, and how it's transferred into proteins, which then has functionality in the cell. So in terms of our DNA here, right, it's our genetic material, so it's what's being copied from one generation to next. This is both in terms of cell function and organism function, right? So Wesley has half of my genetic information and half of my wife's, right? Our cells have all of our genetic information, but when you make a new cell, you need to pass it, right? DNA is composed of 2 DNA nucleotide DNA molecules, right? For paired opposite directions. But they are made up of nucleotides where you have your adenines paired and bonded to a thymine, right? So A to T. And then your guanines are paired to the cytosines, right? And these are G to C, right? So these are complementary bases, right? And so A's only bind to T's and G's only to C's, right? That in DNA, that's it. G's do not bind to T's, C's do not bind to A's. It does not work that way because if you look at it, a double-ringed structure, a nitrogenous base here, binds to a single ring nitrogenous base, right? Via hydrogen bonds. Look at that, right? And A's can only bind to T's because they only have 2 hydrogen bonds available. G's bind to C's because they have 3 hydrogen bonds available, okay? So that's why A's and T's only bind to each other and G's to C's. I will give you an exception to the rule. T's can bind to themselves, and that's bad. That's caused by UV radiation. It causes cancer, skin cancer. It's bad, bad, bad. So in a good, healthy DNA strand, right, the 2 strands are bound together, complementary, anti-parallel to one another, right? They have directionality, A's only bind to T's, and C's only bind with G's. Okay, this is slightly different when we talk about RNA, but we'll talk about that in chapter 3. The structure and sequence of our gene code determines the protein structure. So the order of these nucleotides is really important to determine the structure of a protein. Okay, so these DNA molecules are orientated anti-parallel, so opposite directions to one another, and this helps gene expression occur. So we'll end the chapter there, and I will see you in chapter 3.